while Peter puts up his slides, I'm going to introduce him. So Dr. Peter Kuhn is a Dean's Professor of Biological Sciences and Biomedical Engineering and Aerospace and Mechanical Engineering here at USC. He's also the founder and the co-founder of the USC Michelson Center for Convergent Biosciences. Peter, by training, is a physicist, and this is exactly what, thinking outside the box and bringing people from who aren't necessarily started their careers looking at cancer, but how they can contribute. So his research is, has shed light on how cancer spread through the body, like, I mentioned through the liquid biopsies. He's published over 200 peer-reviewed scientific articles and patents as a result, and he founded Epic Sciences in 2009, which helps develop cancer diagnostic products. And today, Epic is one of the key partners with most biopharmaceutical companies in developing precision ways for diagnosing cancer. So, Peter, looking forward to your talk. It's all yours.
Great. Thank you, Josh. So as I was listening to the last 3 talks, of course, I rapidly reconfigured my talk because this is a fantastic flow of information, and I hope that everybody in the audience is sort of really enjoying this tour as we're going through this. So I will talk about the liquid biopsy as such, and really from the perspective of just picking up from where Elena just left off, of how do we actually implement precision medicine, bridging the gap to enable that continuity of care as the cancer evolves over time. So a lot of this work, of course, is now happening also in the context of post-outbreak, and I just wanted to mention that. Because it is actually important, because it is where a lot of the technologies that we have all built over those past number of years are becoming even more front and center. It is now more clear than ever, I think, that all the work that we have to do in cancer research, it really has to be patient-centric. But it also has to be remote enabled, as we have seen through the various aspects what has happened during the pandemic that has led up to now. And a lot of it has to do with limited access to healthcare facilities, etc. So home health is becoming more and more important. And as we are thinking about liquid biopsies, of course, you can think of mobile phlebotomies as well, and oral drugs being delivered through the mail, as opposed to certain aspects happening in the clinical setting itself. And that really then allows us to help the patient to be prepared and make best use of the interaction with the treating physician in the clinic itself, which is something that we have always talked about, we have always envisioned. It's now just becoming much more front and center. There is this whole discussion around the Internet of Things as it relates to health, and what all we can measure in terms of the health performance of an individual patient. So I will try to wrap this by starting with the patient, talking about the disease itself, the characterization of the disease with the liquid biopsy, and then bring it back with the very last slide, essentially, to really point to this importance of this interplay between the disease and the patient itself. So this is a patient who has really shaped our thought process from very, very early on. This is a lung cancer example. So, as you can see, that PET-CT scan showed a singular nodule in her lungs. And so the decision, because of her frailty, the decision was made to treat this with stereotactic radiosurgery at the time. And when she came back 3 months later for her next imaging session, that primary tumor indeed responded very nicely to radiotherapy. But of course, now she had bone metastasis and mediastinal metastasis. So there's clearly something that we overlooked in this process along the way here. And and she was one of the patients who was early on in one of our liquid biopsy trials, and she had a very large number of tumor cells circulating in her blood at the time of that first at the beginning of her stereotactic radiosurgery. So in hindsight, one has to say, well, wait a minute. In a patient like her, she would have actually been better off with systemic chemotherapy or palliative care, given her frailty and her age. So this is really then something that makes us thoughtful, right? It gives us pause. It tells us, well, wait a minute, how do we actually go about measuring the disease in the human body as we go forward? So the next couple of slides are really meant to just set that stage. So to start with, what is a liquid biopsy? So it's a biospecimen. It's a blood sample. It's a blood sample. For the most part, we're thinking of this as a peripheral blood sample, but can be from various blood sampling throughout the body. It can also be a bone marrow aspirate. It can be a CSF sample. It can be an ascites. Any liquid sample is really something that we call a liquid biopsy. So it's a biospecimen. In each of these biospecimens, there is a heterogeneous mixture of cellular and non-cellular components. So if we think of the blood itself, right, so you have your immune cells in there, the white blood cells, right? You have your red blood cells in there, and then you have all of the plasma, so all of the proteins and the small molecules, the chemicals that flow through the body that give you energy, nutrition, and provide for that defense mechanism. And amongst those, you have, in that whole mixture, you have a small set of very specific cells that we're interested in, right? Those are the cancer cells, or the cells that come from the tumor microenvironment, the proteins that we can measure that are either a direct result of the cancer or an immune response to that cancer. And of course, the nucleotides, meaning the DNA that comes from the cancer itself, that we then try to measure. Now, of course, and it's interesting to just reflect back and ask the question of like, well, where does the science come from, and where are we at today in 2020? So this was actually first discovered by Tom Ashworth in 1868. So it's an old observation during an autopsy where a pathologist recognized that there were cells that in fact looked identical to those from the primary disease were first discovered. And it's that type of a fundamental discovery that really started this whole process of science and this journey of discovery to understand how does the disease evolve in the human body itself, and then how can we actually make use of that. And you know, I used to, when I entered this field, I used to say, we still have not reduced this to practice, but again, thankfully in 2020 we can actually say that we have finally reducing this to practice, and this is becoming useful. But why has it taken so long? So if we just think about, and many of you who are either patients who have been diagnosed or have been diagnosed with cancer at some point, or just have gone to see your doctor for your annual physical, you do blood workup, right? So, and in the blood itself, you then get your white blood cell count. That's typically a couple of thousand per microliters. You get your red blood cell count, which is a couple of million per microliters. And then there's a platelet count in there as well. Now, these white blood cells, so the immune cells, right, they split up into 5 big subtypes. And those are, again, counted in tens to hundreds to thousands per microliters. But the circulating tumor cells that we're now trying to find, they are in the single digits per milliliter. Or even if we look for DNA from the tumor, we're looking at the same level. We're looking at that one copy that reflects that one cell per milliliter of blood, right? So we are now upping the bar in terms of what we have to look for by about 1,000-fold. And that is what makes this just so difficult. And then, of course, these cancer cells, they know how to adapt. So they don't look super different from the immune cells. So for that, it essentially becomes the way we like to think about this is as an extremely fast, high-performance computational game of Where is Waldo? So when you think about this, so this is meant to depict a microscope slide where we would pack about 3 million cells onto here. And the question is, within the 3 million cells that are on here, can we find that 1, 2, or 3 cancer cells, right? Can we actually find Waldo? And the tricky bit about finding Waldo is, of course, that there are lots of either things or others that look just like Waldo, but those are false positives. So the way we have to train up our algorithms, our approaches, is really to separate them out as well as possible. So that's one big challenge. The other big challenge that we always have to be aware of is we have to ask the right question early on. So us as scientists, oftentimes, we fall in love with our tools, we fall in love with our technology because it's really cool. It took us forever to build it. It's very elegant. But I think what we have learned by working with our patients for many years now is really to right away in the beginning of a research project, ask this question about why. What is that clinical context? And I love this example of the caterpillar and the butterfly, because if I ask the question of like, well, which one can fly? Okay. In fact, if I would sequence their genome, their DNA is in fact identical. So that will not answer that question. But I have to look at that proteome, which is what Elena said, the functional readout, right? So that is where those aspects connect, right? So the proteome, of course, will tell me which one can fly. Now, if there would be a pathologist in the room, the pathologist would start laughing and be like, well, Peter, you can also just look at the pictures up here. The one with the wings is likely the one who can actually fly. Okay, so that's why it's always important that we ask this question of what is that clinical context of use. So with that, we get to this, what is the liquid biopsy in 2020? Because we're not anymore in 2005 or 2008 when some of us entered this space, right? So we are now at this level of what we call a third generation liquid biopsy. In this third generation liquid biopsy, we recognize that a tube of blood is just really rich of information. We can find circulating tumor cells. In fact, we can find circulating cells of the tumor microenvironment, which again give us a signal of what's actually going on inside of that tumor. We can find oncosomes and Dolores, who we'll talk after, we will talk about these oncosomes as well. We can then, from these CTCs, we can actually sequence single cells, so we can do the genomic information of single CTCs and of the nucleotides that are floating around, that cell-free DNA that's floating around. We can figure out which is normal cell-free DNA and which is tumor-derived DNA. And now we can do all of this within a single tube of blood. And so that now opens up a whole wide array of opportunities to really participate in clinical care. Now, again, in the beginning, in this research use only framework, but with that goal of advancing it as fast as possible into clinical care, exactly what would be that next step of what Elena just said about her ongoing trial as well. How do we take research forward? So this is just how this workflow essentially works, right? So whether it is a tube of blood, or it is, in fact, we do primary biopsies as well, where we touch that tumor onto glass slides, but we also use bone marrow aspirate, CSF, etc. We then have developed a very standardized approach because we want to compare these different samples. And that's actually a really hard thing to do. If I take a sample out of different parts of the body and try to compare them, put them on the same playing field. So we do that, we cryobank that so we can run along with clinical trials, and then we can do a whole bunch of analysis with that allows us to get to the genomics, which is really the blueprint, but also to the proteomics on the right-hand side, which is that functional readout, right? For those of you who live in the Bay Area, think of the Millennium Tower in San Francisco. That's always I love that example because its blueprint actually looks pretty much okay. It's really hard to find a mistake in that blueprint, but in fact, it's leaning. Right, so its functional readout is actually where you're like, oh, whoa, wait a minute, there's something wrong with the Millennium Tower, okay? So that's why we like that interplay between genome and proteome to really allow us to make progress. I just want to show one example that is really a really important way to think through this. So this is a patient, she was diagnosed in 2012 with primary breast cancer with metastasis to the bone. And she enrolled with us on a clinical trial in 2013, so 7 years ago. But then for the next 4 years, we followed her along this journey of the disease under therapeutic pressure. She had 8 different approaches to a therapeutic intervention. And at some point in 2014, developed liver metastasis. And when we looked at her liquid biopsy results, and what I'm showing in this first slide here is to simply say, oh, okay, interesting, her CTC counts were actually consistent with her other biomarkers. So that was for us a place to be like, okay, this all seems to work. But how do we characterize the disease, right? How do we relate what we are seeing here, not just in saying, disease progression, disease response, but how do we characterize this so that we can relate the treatment modalities to what we actually see in the cancer itself. And this is what we really did here. So for starters, what I just mentioned is that the CTCs as well as the ctDNA, the circulating tumor DNA fraction correlates very nicely with the traditional biomarkers. That was just important to know. But then, of course, we also realized that how interesting her genomic profiles, which we call copy number variation profiles, really inform us about the lineage, the ancestry of her cancer. And so we have done over 500 genomes in her alone. And what we saw in her is actually that she had a pretty stable cancer genome with very few variations, which is why she actually, despite the fairly heavy burden of metastatic disease, actually survived for a long time. It responded to these various therapies. The CTCs that we found early on matched the genomic profile from the tissue biopsies. And interestingly enough, actually, different CTCs we were able to associate with either that primary breast cancer mixed with the bone metastasis, but the clone that really showed up in the liver is actually a distinct clone. And we saw that much earlier than the actual liver metastasis was found that was specific to that liver metastasis. These clones are stable over time. After 3.5 years, here on that right-hand side, a new clone showed up. And that is when really, when therapy failed and that clinical escape happened and the patient passed. And this was really tricky. We're trying to figure out what was going on, and I'll come to that in a minute. The cell-free DNA is because it's a bulk measurement, right? So it's a representation of the aggregate of the most abundant circulating tumor cell clone, which again makes total sense. The ESR1 mutations. So this is really a key, a key readout to understand whether or not ER-targeted therapy works or does not work anymore. Showed up only after that first year, so that did not exist early on. The P53 mutation, again, really interesting, right? Because that tells us really about the genomic stability and the stability of this replication cycle and process. These mutations showed up first, again, now in 2015, so 3 years after the disease was originally diagnosed. And with that, all in all, it was super interesting that the single mutations in that particular cancer were actually quite rare. So her disease was really, for the most part, driven by these large-scale copy number variation changes. And again, that was important to understand how that disease actually progresses over time, and that these single mutations were rare, but then important to start recognizing. The oh, I thought there was one more. Sorry, I forgot there's one more animation that's somehow missing. So this new clone after 3.5 years actually carried a PI3 kinase mutation. And so at the time, neither the Novartis PI3 kinase drug was approved, nor the Roche PI3 kinase drug was approved. So one would imagine in hindsight, right, that if we would have done this in real time, there would have actually been another therapeutic opportunity for that patient, and to see what the disease would have done under that therapy approach. So this is really one of those examples of how we envision how we can manage individual patients over time with the liquid biopsy, because obviously you cannot do primary biopsies 17 times over that time period. And also, obviously, somewhat in quotes, is that because of the heterogeneity of the disease, you really have to think of the complementarity between the primary biopsy and the liquid biopsy as we go forward. There's of course a question of how we translate this, and Josh mentioned this in the beginning, in the introduction, is that this is work by Dr. Howard Scher from Memorial Sloan Kettering Cancer Center in prostate cancer, where he then worked with Epic Sciences, so the company that licensed the technology from the lab, and actually half the lab went over to the startup company at the time when we did that. We take this very serious, right? Because in order to bring these kinds of tools and technologies to the patient, we have to figure out how to commercialize them, how to put them out into companies that can make then that investment to run these large trials that are necessary to demonstrate the benefit. And in this particular case, there is a variation of the androgen receptor. And again, Elena mentioned that androgen receptor that we believe actually plays a role in breast cancer as well, but this is all in prostate cancer, of course. So, and as long as you don't have this variant, which is called androgen receptor variant 7, as long as you're negative for that, you will actually respond quite well to androgen receptor targeted therapy. But if you have this variant number 7, so if you're positive for that, as you can see here, these patients, therapy fails and their survival is really very, very limited. Now that's important to know, but the question is there something that we can actually do about it? And in this particular case, actually, if these patients switch from that targeted therapy over to taxanes, to chemotherapy, they will actually have a much higher chance of surviving, out to an extra 200 days. So it's an important way to really think about how we do this in real time over the course of the disease. So for us, the question now is really in this whole science framework, it's just like, how fast can we repeat this, right? We have now done this in research. We have done this in moving it through into product, moving this into clinical care, because we have to do this for every decision point in cancer as we go forward. So we have built up this entire framework at USC within this Convergent Science Institute in Cancer that really brings two major components together. One is that comprehensive cancer profile with the 3rd generation liquid biopsy approach, together with a whole lot of mathematical oncology, because it's really the mathematicians that then help us bring this data together. And so the mathematics has really become an integral part of how we think about the disease as we go forward. Again, as I said, we have to really, really keep in mind, and that's a whole other lecture. Maybe we do that in a future session like this, where we talk about how the health and the health performance of the patient really plays in with all of this. But that's an important element to this. So with that, I just wanted to finish and say, digitizing the cancer, which is where scientific groups like ours really come into play, because that's where our training is really from. So digitizing the cancer as well as digitizing the health is we believe incredibly important to deconvolute these healthcare disparities. And we have to do cancer research in the context of treatment pressure as well as human individuality to really change the game as we go forward. So with that, of course, a huge thank you to those who are collaborators and funders of the work that we do. And with that, I will finish and hand this back to Josh.
Thanks, Peter. That was great and fascinating. There's a lot of questions coming through, and we'll ask them in just a couple of minutes.